All right, the last thing we need to talk about is the end replication problem. So remember, our chromosomes are linear, so they have two ends. All right, and we have double-stranded DNA. So if one of them is in the 5' to 3' direction, the other is going to be 3' to 5'. And this is just showing one of the ends. The other end would be way over on the left, hundreds of millions of base pairs away. All right? But this end replication problem stems from the fact that when these are split, one of these is going to be in the leading strand and one is going to be the lagging strand. Right? And this is a problem. And in order to show this problem, I'm going to start with a video that really talks about the issue here. All right, so let's watch this video together, and hopefully that'll make sense a little bit better, and then we'll kind of extend from there together.
Ends of linear chromosomes pose unique problems during DNA replication. Because DNA polymerases can only elongate from a free 3' hydroxyl group, the replication machinery builds the lagging strand by a backstitching mechanism. RNA primers provide 3' hydroxyl groups at regular intervals along the lagging strand template. Whereas the leading strand elongates continuously in the 5' to 3' direction all the way to the end of the template, the lagging strand stops short of the end. Even if a final RNA primer were built at the very end of the chromosome, the lagging strand still would not be complete. The final primer would provide a 3' hydroxyl group to synthesize DNA, but the primers would later need to be removed. The 3' hydroxyl groups on adjacent DNA fragments provide starting places for replacing the RNA with DNA.
So remember, that's DNA polymerase 1 that is extending from the strand that DNA polymerase 3 already made, but it needs that 3' OH group. Without a 3' OH group to start, there is nothing to use for DNA polymerase 1 to replace the RNA primer. So this is where our RNA primer was. And in order to replace that, I would have needed something over here with that 3' OH group, because it can only grow from 5' to 3', so from right to left. So there's no way to replace that RNA primer in this case.
However, at the end of the chromosome, there is no 3' hydroxyl group available to prime DNA synthesis. Because of this inability to replicate the ends, chromosomes would progressively shorten during each replication cycle.
All right, that last statement there is key. So because we have that RNA primer that can't be replaced, right? Because we have that RNA primer that can't be replaced, that is eventually going to degrade, and our body does not like single-stranded DNA, so it's actually going to chop that up. So what happens is that after every series of replication, the ends of the chromosomes are going to get slightly smaller. They're going to lose that little bit because of that primer that could not get replaced, that was RNA, that gets degraded. So again, the end replication problem is that the ends of the chromosomes get slightly shorter every time of replication. Right? And obviously this is a problem. This is a problem because if you're losing part of that genetic information when the DNA is replicated, the new cells aren't going to have the full set of information. That was the reason for replicating it in the first place. So, this is what you really need to understand. What are the 3 ways that nature gets around this? Because this would be a big problem. Well, if you are a prokaryote, you get around that because bacteria have circular DNA. Their chromosomes are full circles. So bacteria get around this problem because they don't have ends. Therefore, they don't have the end replication problem because their genome is circular. All right? However, eukaryotic cells with those long linear chromosomes do have the ends, and that's a problem. So there are 2 things that eukaryotic cells can do. One is eukaryotic cells have telomeres. Right? And so if we look down here, we see that we have this repeated sequence, GGG, TTA, GGG, TTA, GGG, TTA. So these are telomeres. These are long, long repeats of those at the ends of the chromosomes. Well, why? That sounds like nonsense. Well, there it is. That is why. If you add kind of this non-important, nonsensical repeat DNA at the end of the chromosomes in these telomeres, even if they get shorter by a little bit each time, you have a long buffer zone before they start to get to the real important genetic information. So these telomeres are kind of just extra repeats at the end, so that as they get shorter, you're not losing key genetic information. But this is also why cells also have a limited number of times they can divide. Because eventually, after dividing, 50, 60, 70 times, even those telomeres are gone, and now if you start to lose key genetic information that was inside those telomeres and the other parts of the chromosomes, then that cell is going to have problems and dies. All right? So, 2 solutions so far. Prokaryotes don't have to worry about it because they have circular DNA. Eukaryotes have these telomere repeats at the end to give them a buffer where when they're being shortened a little bit by a little bit, it takes a long time to get to information that actually is detrimental. However, there are certain cells that have to divide a lot more times than that would allow. All right, so these are things like gametes, stem cells, the sex cells, anything that has to divide a lot are basically that's not good enough, right? So these have an enzyme called telomerase. And I'll give you the take-home message of telomerase first, and then we'll see how that happens. Basically, telomerase takes those repeats and extends them right before each replication. It extends them basically the same amount that they will get shortened. So telomerase extends the telomeres the same amount so that they get shortened, so the net is no loss. Right? So telomeres, again, extends the telomeres so that when they get shorter, you really haven't lost anything because they extend them again right before they're shortened. Extend again right before they're shortened. So these can actually divide infinitely without losing the key information because they will actually never shorten past the telomeres. All right, so how does this happen? Let's go back to our video.
Each replication cycle. This end replication problem is solved by the enzyme telomerase. The ends of chromosomes contain a G-rich series of repeats, called a telomere. Telomerase recognizes the tip of an existing repeat sequence. Using an RNA template within the enzyme, telomerase elongates the parental strand in the 5' to 3' direction, and adds additional repeats as it moves down the parental strand. The lagging strand is then completed by DNA polymerase alpha, which carries a DNA primase as one of its subunits. In this way, the original information at the ends of linear chromosomes is completely copied in the new DNA.
All right, so what that's basically showing you is that telomerase basically has this template sequence that mirrors the repeated sequence of our telomeres. And so its job basically is to extend these telomeres out. So it binds to this TTA here, and then you can see we have a template. Next to the C will be a G. We'll end up with G, 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 T, T, A. And that there just extended that telomere by 1 repeat. And it can move over and it can do this again. So basically it extends that telomere out, and now when the primer has to bind here and to replicate this and the primer is gone, well, yes, you'll lose a little bit, but you only lose the same amount of telomere that you already added. So that's what telomerase does. It extends the telomere the same amount that it basically will be lost. And that's how these cells can replicate forever. So the question then is, which cells will replicate forever? So here you see it kind of extending it, getting more telomeres, and then the RNA binds to it, extends this here, and now basically you get the degradation, but only lose the amount that you added. So which of the following cells are likely to have active telomerase gene? Bacterial cells, because they need to divide an unlimited number of times. Well, yes, bacteria do need to divide an unlimited number of times. That's how they stay alive and keep growing. But they don't have to worry about telomerase. Why? Bacteria have a circular genome. They already solved that by the circular genome. Neurons, they are vitally important to our body, but they don't divide much. The neurons you have as an adult are pretty much the ones you have, so they're not dividing much. They don't need telomerase. The telomeres are enough. Human somatic cells that normally divide about 50 times before they undergo cell program death, well, that also is just the telomeres. The telomeres are enough of a buffer to allow them to divide those 50 times. These are kind of normal for our cells. However, stem cells that have to grow and divide many, many times and regenerate themselves to all the cells in your body, If they have to divide a lot, they would need telomerase. So in summary, we have the end replication problem because when the primer has to bind to do the lagging strand at the very end, that little bit, that primer can't be replaced, and you lose a little bit of the end of the telomere, of the end of the chromosomes. All right, that's the problem. The three solutions are: bacteria don't have to worry about it because they have a circular chromosome. Eukaryotic cells have telomeres, these repeats at the end, and that gives it a buffer zone so that as it loses some ends of the chromosomes, you're not really losing vital information for at least 50 replications. And then for cells that need to divide a lot, telomerase actually extends the ends the same amount that they will be reduced later on. All right, thank you.